Well, good evening. This is Miss Dagenford, and I hope you're enjoying your 4-day weekend. We are going to watch a video that will hopefully get us started on Monday and help us to be prepared. And so I wanted to start with the objectives. You should be able to identify the molecules that constitute the cell membrane. You should be able to describe the complex structure of the cell membrane. And then ultimately, we want to describe the movement of molecules through the membrane. So let's get started. And what I did is I divided it into these main points. Now you will get these pictures as a practice and review. So the important thing is to draw some of the things that I draw. Don't try to print this out. You will get it. Now, but we're going to start with is the cell membrane is made of phospholipid molecules. Now, if you kind of break down this word, phospho, and then we have lipids. So there are some things that we have already covered. Phosphates and lipids we talked briefly about. So what does this molecule look like? Well, a phospholipid has two distinct parts. It has two fatty acid tails, and the tails kind of dangle down. And hopefully, if you give it a quick look, there's something that's interesting about this one on the right. I'm going to go ahead and circle that double bond. So in your mind, hopefully you are thinking that it is an unsaturated fatty acid, and the one on the right is a saturated fatty acid. Now, there are some interesting things that these affect, but for now, just know that that should be something that you remember. There's a couple of interesting things about these. This is nonpolar. It does not mix with water. And we call this hydrophobic. And you might recall that phobia refers to fearing. So the other name for this is water fearing. Now that's a bit of a misnomer. It's not that it repels or it's repulsing water. It just doesn't mix. It's neutral. Remember, if you had water with the partial charges, negative, partial negative, and partial positive, that water molecule is going to be attracted. Here's our positive charge. We've got a negative charge. We've got some partial charges going on. That water molecule is going to be very nice and attracted to this top part. So this top part is polar. And the other word we use is hydrophilic. And hydrophilic refers to water loving. You might think, the city of brotherly love, Philadelphia. So philic is a Greek term for brotherly love. So those are our two main parts. And if you notice, if I outline it, if I outline this, you kind of get this little structure. And in fact, there's our little model at the bottom. So from now on, we can kind of just draw a phospholipid as looking like that, with our two nonpolar tails and the one polar head. Now, let's go ahead and start thinking about what this means for its behavior. If we have this beaker of water, and our beaker of water is nice and spread out, and into our beaker of water, we place phospholipids. We just got a test tube of them. We're going to pour it in, and at first these phospholipid molecules are all mixing. Now remember, nonpolar, what was the molecule in class that was nonpolar? Oil. So this bottom section is similar to oil. And we already talked about in class what happens when we mix water and oil. Well, salad dressing, you shake the bottle up and then you leave it on the counter and they separate. So usually if I ask students to ponder what happens after a few minutes of letting these things settle, what is it going to look like? And I usually get a couple of answers, and they're both very intuitive, very nicely done. If you are thinking, oh, these nonpolar tails are going to stick up. That's one possibility, just like oil and water. Now, another possibility is that the oil droplets in the oil and water, you might think, oh, okay, well, maybe the phospholipids are going to form something similar where all of the nonpolar tails are facing in and then the polar heads are facing out. And of course, this is a limitation. This is a two-dimensional drawing. This would be a sphere, and you would be correct. Now, what that means is that the cell membrane has a very distinctive orientation, and this is due to the chemistry. So I'm just going to go ahead and draw, and I would like you to do this as well. We're going to go ahead and draw 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then I'm going to put 10 on the bottom. Now, this only works if we're stipulating that the phospholipids are surrounded by water. So here's our nonpolar tails. And each polar head has two nonpolar tails. And I also want to indicate that this is going to keep going. But of course, I don't want to keep drawing that, and that's a bit of a burden. And the water would be facing each side. Now, I'm hoping you're, and we're going to talk about this on Monday. So let's think about that. We have all of these nonpolar tails, nonpolar fatty acid tails. So this inner part ends up being like oil, very much so. And in fact, a lot of times I just say, go ahead and think about this as kind of being this mixture of oil. And that really helps us think about this being a barrier. We're creating a barrier, and that's what a cell membrane is. It lets some things in, it keeps things out. And then on the outside, we have our non-polar. I'm sorry, outside would be polar. And then the inside is non-polar. Now, this is something that's very important. We're going to practice this, and we're going to make sure we understand the right orientation. All right, so now on to the next one. Now, the cell membrane is a complex mixture of the molecules, the phospholipid molecules, and proteins. Those are the bulk of the membrane, and then followed by carbohydrates and steroids. So let's take a quick look at this rather nicely drawn artistic model. And just as before, we can identify here's that oil layer all in the middle. Anything that's teal, that's going to be that oil layer. And then on the outside, that's going to be, and so with oil, my bad. So the oil would be our non-polar section. There's non-polar tails, and then the polar would be on the outside. I'm just going to go ahead and identify that. So these would be polar, and that would be polar. And this is assuming in this diagram we'd have water on the outside, water on the inside. And just to give you a sense of perspective, the cells that we drew, like the amoeba, the line that we would see on the outside, that is our cell membrane. And then we'd have our nucleus. So imagine this part, we're just taking this little section of the cell membrane and we're blowing it up. We're kind of expanding it to see what it looks like, and that's what we would see. So here's one other picture, one other artistic rendering. And the part that I like here is you kind of get a sense of these proteins as kind of being that little twisty guy that I showed you in class, little bendy shape. That's not the only thing we have. We have side chains of sugars. We have steroid molecules, cholesterol, and we'll talk about those on Monday a little bit. They do perform very important functions, but overwhelmingly the structure is made of the phospholipids and proteins. Now, this presents an interesting condition for the cell. This ends up being a nice barrier, but because it's a barrier, that means things can be blocked. The cell membrane is selectively permeable. Now, selectively is somewhat Obviously, we kind of know, okay, it's selective. It's only going to let some things through. And that's what permeable means. Permeate. If I said that perfume permeated the air, it spread. And so permeable means that some things can come in, and selectively means that only some things can get in. Now you want to think about, remember, this is oil. This is like oil on the inside. And so remember our little stipulation in the previous unit, polar and non-polar. Let me go ahead, non-polar. Do not mix. And remember, we can say polar would be water, non-polar would be oil. Oil and water don't mix. So anything that is polar or charged is not going to mix with those non-polar tails. So we can go ahead and make our list. We can make, okay, anything with charges. Well, that's sodium with a positive charge, chlorine with a positive charge, potassium with a positive charge. They will not mix. Now, mixing also can be related to passing. They cannot pass through. So anything that's charged or anything that's polar cannot pass through those phospholipid molecules. Now, sometimes they can pass through, but they're very slow. It's going to happen pretty rarely. So if we go into our diagram, here is our charge. And notice the arrow is indicating that they can't go through. Here's another set of charged, positive and negative charge, and they can't go through. So, they're going to, and again, it's because they can't mix. So they're going to come floating up. They're just going to sit there, and then because they're moving, they'll just bounce off. Now here's one that's non-charged. So non-polar molecules can pass through. Because think about it, it's just like oil and oil. It's going to mix. Now, what are some examples of each of these? So an example of charged or polar, charged would be sodium, potassium, chlorine ions, and water. Now, water is an interesting case. It can go through, but it's pretty polar, so it's pretty slow. It doesn't happen that readily. And then non-polar can pass through. So what are some examples of that? Well, we have oxygen, carbon dioxide, carbon monoxide, unfortunately. They can go through. And so that gets us started for the next thing that we're going to talk about, which is the diffusion.